All right, we're going to start today by talking about what led up to the discovery of the structure of DNA. So there's a guy named Frederick Griffith who did his work in 1928, well, the work that we're going to talk about in 1928. And his deal was that he was trying to figure out how bacteria cause pneumonia. So he wanted to say, you know, people were having pneumonia, they knew it was bacteria, but they didn't know how or what was going on. So what he did was he had these bacteria that were disease-causing, and then he had other bacteria that were harmless, that were not disease-causing. And so what he did was he took the ones that were disease-causing and he killed them. And then he took these dead bacteria and he combined them with the other ones that were alive and they were harmless ones. So killed the bad ones, put them in with the live good guys. And what he found out was that those live good guys suddenly became disease-causing bacteria. Dun, dun, dun. He called that transformation because what happened basically is that the good guys transformed into bad guys. So that was our first sort of foray into figuring out what the heck is going on here. So then we've got Oswald Avery, who was Canadian, and his famous study was in 1944. He was working with other scientists in New York, and what he was trying to do was replicate Griffith's experiment and figure out which molecule specifically it was that caused the transformation. So Griffiths figured out the transformation happened. Oswald Avery was like, hey, but what is it? What molecule is doing that? So what he did was he destroyed everything in a cell that wasn't DNA. So he destroyed all the proteins, he destroyed all the carbohydrates, he destroyed all the lipids. He was left with just DNA. And what he found was that transformations still happened. So his conclusion then was if DNA is all I gave him, then DNA has to be the thing that's making this change. 1952, we get to Alfred Hershey and Martha Chase. They were Americans, and they worked with these things called bacteriophages. And bacteriophages, if you look at that word, we know bacteria refers to bacteria, and phage means eating. So they are viruses that attack bacteria. So they are bacteria-eating viruses, basically. So they took bacteriophages, which are really just viruses. So for the most part, what they are is DNA inside a protein coat. So if you think of an M&M as chocolate in a candy coating, a bacteriophage is basically DNA in a protein coating. What they did was they marked the proteins with radioactive sulfur, and they marked the DNA with radioactive phosphorus. And the thing is, radioactive sulfur and radioactive phosphorus are going to glow in different colors. And so they let the viruses infect the bacteria. So they took these bacteriophages, they let them infect the bacteria, and then they tested the bacteria. And what they found was that the only glowing they could see was the glowing color that matched the radioactive phosphorus. So their conclusion then was that DNA is what was carrying the genetic info. So when these bacteriophages were inserting something into the bacteria, it was the DNA that they were inserting. Erwin Chargaff, another American, 1951 and 52 was when he was doing his work. Everybody already knew that DNA had two different kinds of molecules in them. There were purines, which are the two specific purines are adenine and guanine. And then there are pyrimidines, which are cytosine and thymine. If you just need a quick way to remember which is which, and it's not super important that you remember which is which, but pyrimidine has the letter Y in it, and both cytosine and thymine have the letter Y in them as well. So pyrimidines with a Y are cytosine and thymine, which also have a Y. But again, it's not super important that you remember which is which. So the purines are adenine and guanine, pyrimidines are cytosine and thymine. So what Chargaff figured out with doing his research is that the amount of adenine in a DNA molecule is equal to the amount of thymine. And then he also found out that the amount of cytosine in a DNA molecule is equal to the amount of guanine in a DNA molecule. So what he figured out was that the amounts were equal. Okay, so another way that we can write those amounts, so concentration, we write with these brackets, with the big square brackets that look like parentheses, but they're squared off instead of curvy. Okay, so the concentration of adenine is the same as the concentration of thymine in DNA. and the concentration of cytosine is going to be equal to the concentration of guanine. So those are Chargaff's rules. The question, though, was why? Why did that happen? Okay, so enter Rosalind Franklin, and then we know that at the same lab, Maurice Wilkins was working, and Raymond Gosling, who was a lab assistant and also a student who was working with them. So Rosalind Franklin shot X-rays at DNA. This was her job. It's called X-ray crystallography. And so she would shoot X-rays at things and then get these pictures, basically. She would shoot X-rays at crystals and get pictures. And she shot X-rays at the DNA, and she analyzed the pattern that they made when they bounced back off of the DNA. So this is an example of the kind of picture that Rosalind Franklin took. And this is actually a really, really famous one that was kind of helpful in the breakthrough, or integral in the breakthrough to what's the actual shape of DNA. The fact that there was an X-shaped pattern right here indicated that there were two strands that were wrapped around each other. We call that a helix. So a helix refers to the fact that you have two strands wrapped around each other. And then the angle of the X shape indicates that there were two strands as opposed to 3 strands or 4 strands or, you know, something else. So all kinds of crazy math went into figuring that out. Okay, there were other clues in there. I don't know, I don't do X-ray crystallography. There were other clues that indicated that the bases, adenine, guanine, cytosine, and thymine, were actually on the inside of the helix. Okay, so her photographs were super duper important to figuring out what was going on and how this structurally, how DNA structurally was set up. So Watson and Crick, Watson American, Francis Crick British, in 1953, first experimented unsuccessfully with cardboard and wire to make models. But then they saw images of Rosalind Franklin's X-rays, and that gave them enough clues to lead them to a more successful model, and they got the lab to actually build legitimate parts for them, so they didn't have to use cardboard and wire anymore. And so their model showed a double helix, so 2 strands wound around each other. So basically what Rosalind Franklin's photos showed, but they built a model, okay? And people really like models, and they really like to look at models. So if you look at the background of this picture, you can see a model of a double helix. Okay, so what does it look like? So we're going to start with a phosphate and a deoxyribose backbone. Don't panic. That's just big words, but it's no big deal. So this phosphate group is here. I've drawn it as a circle. It's not really circle shaped, but I've drawn it as a circle here. And deoxyribose is a 5-carbon sugar, okay? It's a 5-carbon sugar, so it looks like a pentagon. These are not all carbons. One of these corners is an oxygen, and then you have another carbon out here, but that's just a fun fact. So you have a 5-carbon sugar and a phosphate, and it's an alternating sugar-phosphate backbone. So you have deoxyribose attached to a phosphate, attached to a deoxyribose, attached to a phosphate, attached to a deoxyribose, attached to a phosphate, deoxyribose, phosphate. So you've constantly got sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way up this backbone. And then on the inside, you have these things called nitrogenous bases. Nitrogenous bases, I've already told you the names of them: adenine, guanine, cytosine, and thymine. Quick check, which 2 of these are purines? I hope you said adenine and guanine. So which 2 of these are pyrimidines? Hopefully you said cytosine and thymine. All right, so we have adenine here and guanine here, and notice they're kind of big. And then we have cytosine and thymine, and notice that they are comparatively kind of small, okay? That's because purines are bigger. They're a double ring structure, and pyrimidines are a single ring structure, okay? So you have these bigger purines and these smaller pyrimidines, but adenine, guanine, cytosine, and thymine are your 4 nitrogenous bases. You have to know those, okay? So again, we've got our sugar-phosphate backbone, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, and then we have our bases. And notice that our bases are attached to the sugars. None of these bases are attached to the phosphates. The bases attach to the sugars, okay? And then there's a second strand that runs in the opposite direction of the original strand. So notice that while this goes phosphate sugar, this one goes sugar phosphate. Okay, and you could kind of say that these are upside down, but molecules don't really have an upside down. They're sort of in 3D space and whatever. But again, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. But here we have sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. But notice that still the bases are all attached to the sugars. And then in the center, the base is attached to each other. Now notice how this spacing works. You have a 2-ring structure attached to a 1-ring structure, so you have 3 rings altogether. Down here you have a 1-ring structure attached to a 2-ring structure, so again you have 3 rings altogether. So that keeps the spacing 3 rings, 3 rings, 3 rings, 3 rings. Okay, so adenine bonds to thymine and cytosine bonds to guanine, and we know that because it's an inference based off of Chargaff's rules. Now, we have since gone back and proven it through all kinds of other like radioactive tagging and things, but we figured it out because of Chargaff's rules. Because if the amount of adenine is equal to the amount of thymine, then it makes sense that every time there's an adenine, there must be a thymine attached to it. If the amount of guanine is equal to the amount of cytosine, it makes sense then that everywhere there's a guanine, there must be a cytosine attached to it. Now, if you're having trouble with the bonding rules, remembering that A bonds to T and C bonds to G, again, I'm a very visual person, so the way I remember this is that C is a curvy letter and G is a curvy letter. So the curvy letters bond to each other. A is a very pointy letter and T is a very pointy letter, so the pointy letters bond to each other. Okay, so adenine bonds to thymine and cytosine bonds to guanine. So we have these 2 strands running opposite to each other. They're attached with the bases in the middle, and then the last step is we just twist, and that's where we get our helix shape. And that is the shape of the double helix.